Aseptic environments are the backbone of pharmaceutical manufacturing, healthcare, and laboratory operations. These highly controlled spaces exist for one reason: to prevent microbial contamination where even the smallest failure can compromise patient safety, product efficacy, or critical research. Maintaining sterility in these environments isn't optional, it's essential. Aseptic environments are tightly controlled spaces designed to eliminate microbial presence. They are used in pharmaceutical production, research laboratories, operating rooms, and healthcare facilities. Anywhere sterility is a requirement, not a preference. These environments rely on multiple safeguards working together, including controlled airflow, pressure differentials, and high-efficiency filtration systems like HEPA filters. Strict regulatory standards from organizations such as the FDA, EMA, and WHO define how these spaces must operate to protect both products and people. One microbial contaminant can result in product recalls, patient harm, regulatory action, and loss of trust. Maintaining aseptic conditions is both a regulatory responsibility and a moral obligation. Sterilization is the foundation of aseptic control. It ensures that surfaces, tools, equipment, and air are free of viable microorganisms capable of compromising sterility. True sterilization isn't just about cleanliness, it's about reducing the probability of surviving microorganisms to an extremely low level. This is especially critical for injectable drugs, implantable devices, and other products that directly interact with the human body. From reusable tools to single-use components and ISO Class 5 critical zones, sterilization underpins every aspect of aseptic processing and protects both product integrity and organizational reputation. Different environments and materials require different sterilization approaches. Steam sterilization, or autoclaving, is widely used for heat-resistant materials. Dry heat is applied where moisture-sensitive materials are involved. Chemical sterilization, including hydrogen peroxide and ethylene oxide, allows penetration into complex geometries. Radiation sterilization, such as gamma or electron beam, is often used for pre-packaged sterile products. Each method must be precisely validated, meeting defined parameters for time, temperature, dose, and distribution, using biological indicators and sterility testing to ensure effectiveness without damaging materials. Sterilization is not without risk. Human error, equipment failure, and complex system geometries can all compromise outcomes. Environmental conditions like humidity can reduce effectiveness, while some chemical sterilants pose safety and environmental concerns. These challenges demand rigorous validation, disciplined procedures, and continued innovation. Modern sterilization technologies are advancing rapidly to meet today's aseptic demands. Vaporized hydrogen peroxide systems have become a preferred alternative due to their ability to evenly decontaminate entire spaces. Building on this, Curis hybrid hydrogen peroxide (HHP) technology delivers high-level efficacy using a low-concentration solution, improving safety, material compatibility, and cycle efficiency. Automation reduces human intervention, while real-time monitoring, advanced biological indicators, AI-driven analytics, and predictive maintenance further strengthen reliability and compliance. These innovations are closing gaps in traditional sterilization and enabling continuous aseptic integrity. Precise sterilization in aseptic environments requires advanced technology, rigorous protocols, and a shared commitment to excellence. Manufacturers, technicians, regulators, and innovators all play a role in protecting public health and advancing science. Explore how Curis system supports safer, more effective aseptic environments and take the next step toward precision-driven sterility assurance.